Welcome back to the deep dive. Today we've got a really specific focus just for you, especially if you're gearing up for Dr. Remy's Biosci 221 Microbiology unit, #1B quiz over at College of the Canyons. We're skipping the fluff.
Exactly. We're drilling right down into the essentials from chapters 3 and 4, eukaryotes, viruses, prions, things like structures, how they're transmitted, how we diagnose them.
Basically, the stuff you really need to know as a future nurse.
That's the idea. Think of it as understanding the why behind it all. Why does this drug work on fungi? Why does that parasite need a cyst to spread?
Love it. So the plan is start with the big picture, eukaryotes versus prokaryotes, especially for drug targets, then jump into protozoa and helminths, how they survive and spread.
And finish up with viruses, their life cycles, the enzymes they sometimes carry, how they evolve, and how we actually detect them in the lab.
All right, let's dive straight in. No time to waste. Part 1, eukaryotic fundamentals.
Okay, so when you look at microbes, the really fundamental split is prokaryotes, like bacteria, and eukaryotes, fungi, protozoa. This difference is like the bedrock of treatment strategy, isn't it?
Absolutely. It all comes down to selective toxicity. How do we kill the microbe without hurting the patient?
Right.
And there are 3 main differences we exploit. First, where the DNA lives. Eukaryotes have that nice membrane-bound nucleus with linear chromosomes. Prokaryotes, their DNA, usually circular, is just kind of bundled in the nucleoid region. No membrane.
And eukaryotes are just generally more complex inside.
Yeah, they've got all those organelles, mitochondria, Golgi, all wrapped in membranes. Bacteria don't have that internal compartmentalization. But maybe the most critical difference for some drugs is the ribosomes.
Ah, the protein factories.
Exactly. Eukaryotes use the bigger 80S ribosomes. Prokaryotes use smaller 70S ribosomes. That size difference lets some antibiotics just shut down bacterial protein making while leaving our own 80S ribosomes alone. It's really quite elegant. Okay, that works for bacteria, but fungi, they're eukaryotes, just like our cells. So how do we find that selective toxicity against them? What's different?
Good question. You have to find targets that fungal cells have, but human cells don't. And luckily, fungi offer up a couple of great ones. First, there's cell wall.
Which we don't have at all.
Right. Fungal walls have chitin and also beta-glucans. So drugs like the echinocandins, they specifically mess up the enzyme that makes a key glucan, beta-1,3-D-glucan. No wall synthesis, the fungus is in trouble.
Okay, the wall is one target. What's the other big one?
The cell membrane. This is like the prime real estate for antifungal drugs. See, fungal membranes rely on ergosterol for stability. Our membranes use cholesterol.
Ah, that structural difference again.
Precisely. And we hit it 2 ways. The azoles, they inhibit the synthesis of ergosterol. Less ergosterol, the membrane gets leaky and unstable.
And the second way? You mentioned the heavy hitters earlier.
Right, that's the polyenes, and the classic example is amphotericin B. It doesn't block synthesis, it binds directly to the ergosterol that's already there, and when it binds, it forms pores, like little holes, in the membrane.
So everything just leaks out?
Pretty much. It's quite potent, but that ergosterol difference is absolutely key for understanding why it works.
We should probably quickly touch on the different forms fungi take. Molds versus yeasts?
Sure. Yeasts are basically single-celled fungi. They usually reproduce by budding. Think Candida albicans, a common opportunistic yeast.
Right. Part of our normal microbiota sometimes, but can cause trouble.
Exactly. Molds, on the other hand, they grow as these long, thread-like filaments called hyphae. A whole network of hyphae is a mycelium. That's the fuzzy stuff you see.
And then there are the ones that switch.
Yeah, the dimorphic fungi. These are really interesting. They're typically a mold out in the cooler environment, but when they infect a human, the higher body temperature triggers them to change into a yeast form, or sometimes a structure called a spherule. Histoplasma is a classic example.
Before we leave fungi, we absolutely have to mention Candida auris. It keeps popping up in the news.
Ugh, C. auris. Yeah, it's a major headache in healthcare. It's an emerging yeast, causes invasive infections, but the real kicker is its multi-drug resistance. It's often resistant to multiple classes of antifungals.
And it spreads easily in hospitals.
Terribly easily. It sticks at surfaces. It can colonize patients' skin, healthcare workers' hands. It just spreads really effectively.
So if you suspect it, what's the key thing for an RN?
Immediate, strict contact precautions, like non-negotiable, and tell the lab. Standard yeast ID methods might misidentify it. You often need specialized techniques like MALDI-TOF, that's a rapid protein analysis, or PCR to be sure. It really highlights the need for thorough environmental cleaning too.
Okay, Shifting gears now from fungi to protozoa. Still eukaryotes, still single-celled, but their survival often depends on switching between two life stages, right?
Yeah, that's a critical concept. You got the trophozoite and the cyst.
Trophozoa is the active one.
Right. That's the feeding, multiplying, metabolically active stage. It's the form that's actually causing the disease inside the host, but it's usually quite fragile outside the body.
So that's where the cyst comes in.
Exactly. The cyst is the dormant survival stage. It forms a protective wall, shuts down metabolism, and it's resistant to drying, chemicals, stomach acid. This is the form that typically transmits the infection, often through contaminated food or water.
So the process is encystment to form the cyst, then excistation back to the trophozoa once it's ingested.
Precisely. Encystment happens usually as it passes out of the host. Excistation happens when the cyst gets into a new host.
Okay, let's connect some key protozoa to their transmission. How about Giardia?
Giardia duodenalis causes giardiasis, that persistent diarrhea. Classic fecal-oral transmission via ingestion of cysts in contaminated water or food. Diagnosis is often straightforward with stool antigen tests now.
And malaria. Plasmodium falciparum. Totally different route.
Completely different. No cysts involved in transmission to humans. The Anopheles mosquito injects sporozoites directly into the bloodstream when it bites. Diagnosis relies on rapid tests or seeing those characteristic ring-formed trophozoites inside red blood cells on a blood smear.
Then there's Toxoplasma gondii. That one is a bit more complex, isn't it? Especially concerning pregnancy.
Yeah, coxoplasmosis is tricky because you can get it two main ways. You can ingest the oocysts, which are shed in cat feces and contaminate soil or water, or you can eat undercooked meat containing dormant tissue cysts. The big concern is congenital infection if a mother acquires it during pregnancy.
And diagnosis is usually blood tests.
Mostly serologic testing, yeah. Looking for antibodies to see if someone's been exposed.
And we need to mention Naegleria fowleri, the brain-eating amoeba, especially with climate change.
Right. Naegleria is terrifying. It causes primary amoebic meningoencephalitis, or PM, which is almost always fatal. The key thing here is transmission. The trophozoite stage infects humans by entering the nasal passages, usually during swimming or diving in warm freshwater lakes or rivers.
And warming waters are expanding its range northward.
Unfortunately, yes. We're seeing cases further north than historically reported. It's a direct link between environmental change and infectious disease risk.
Okay, let's switch from single-celled protozoa to the multicellular worms, the helminths. Big difference here, right? You can often see the adults.
You can, although diagnosis usually relies on finding microscopic eggs or larvae in samples. But the single most important clinical point about helminths:
What's that?
adult worms do not multiply inside the human host. If you swallow 10 infectious larvae, you end up with 10 adult worms. They don't reproduce to make more adult worms within you.
So the severity of disease depends on how many you acquired initially.
Exactly. The worm burden is key. Unlike bacteria or viruses or protozoa that replicate exponentially inside you.
Let's use the pork tapeworm, Teunia solium, as an example. It causes two very different diseases depending on what you swallow.
Right. If you eat undercooked pork containing the larval cysts, the cysticerci, you get toniasis. The adult tapeworm lives fairly harmlessly in your intestine. Annoying, but usually not dangerous.
But if you swallow the eggs...
That's the disaster scenario. If you ingest the eggs, maybe from food contaminated by someone who has the intestinal tapeworm, the larva hatch, get into your bloodstream and travel. If they end up in the brain, they form cysts there, causing neurocysticercosis. That leads to seizures, neurological problems. It can be very serious.
Wow, huge difference based on life stage ingested.
Massive difference.
And quickly, the most common worm infection in the U.S., pinworm.
Yep. Anaerobius formicularis.
Wow.
Causes that intense perianal itching, especially at night. Transmission is super easy. The eggs get onto hands, under fingernails, onto bedding, toys.
Yeah.
And they become infective within hours.
Diagnosis is the famous Stotch tape test.
Still the classic way, yes. Pressing tape to the perianal skin in the morning to pick up eggs the female laid overnight.
One more helminth group, hookworms. Cause anemia, right?
Yes. Necutor americanus and Ancylostoma duodenale. They live in the intestine and feed on blood, leading to iron deficiency anemia. The key transmission here is skin penetration. Larvae in contaminated soil burrows directly through bare skin, often the feet. Diagnosis is finding the eggs in stool.
All right, moving on to the acellular world, viruses. They're basically just genetic material in a protein coat hijacking our cells. But some need to pack extra tools, don't they? Which ones need their own special polymerase?
The negative-sense single-stranded RNA viruses, or ssRNA viruses. Think influenza, Ebola, rabies, RSV. Their RNA genome is like the photographic negative of messenger RNA. Our ribosomes can't read it directly.
So they have to bring the enzyme to make the positive copy.
Exactly. They must package their own RNA-dependent RNA polymerase, RDRP, inside the variant. That enzyme reads the negative sense genome and makes positive sense mRNA copies that the host cell can translate into viral proteins. No RDRP, no replication.
Okay, and the other group famous for bringing their own enzymes, retroviruses.
Right. Group C's viruses, like HIV. They pack two critical, unique enzymes. First, reverse transcriptase. It does something amazing. It reads their RNA genome and makes a DNA copy.
RNA to DNA, backwards from the usual flow.
Totally backwards. That's why it's reverse. And second, they carry integrase. Once the DNA copy is made, integrase is the enzyme that literally cuts the host cell's chromosome and pastes the viral DNA copy into it.
And that integrated viral DNA is called?
A provirus. It becomes a permanent part of the host cell's genome.
Let's talk structure. Big difference between enveloped and non-enveloped viruses. Why does that matter for, say, disinfection in a hospital?
Oh, it matters hugely. Enveloped viruses like influenza, HIV, SARS-CoV-2, herpes viruses, they steal a piece of host cell membrane as they exit the cell. That lipid envelope makes them relatively fragile.
WIMPs, you called them earlier.
Yeah, basically. Soaps, detergents, alcohols, standard disinfectants, they easily disrupt that lipid envelope and inactivate the virus. They don't survive well on dry surfaces.
But non-enveloped are tougher.
Much tougher. Non-enveloped or naked viruses, think norovirus, rotavirus, adenovirus, HPV, only have their protein coat, the capsid. Proteins are much more resistant to drying, detergents, alcohol, and many common disinfectants. You often need stronger agents like bleach solutions to reliably kill them.
So knowing enveloped versus non-enveloped directly impacts your cleaning protocol.
Absolutely. Huge implications for infection control.
Let's quickly touch on bacteriophages, viruses that infect bacteria. They have two main life cycles, right? A lytic and lysogenic.
Correct. Virulent phages only do the lytic cycle. They infect, replicate rapidly, make lots of new phages, and then boost or lyse the bacterial cell, releasing the progeny. Quick in, quick out, cell death.
But temperate phages have an option.
Right. Temperate phages can choose. They can go legolytic, or they can enter the lysogenic cycle. In lysogeny, the phage DNA integrates into the bacterial chromosome.
Or it's called a prophage.
Exactly. As a prophage, it just passively replicates along with the bacterial DNA every time the bacterium divides. It can stay dormant like that for a long time. But environmental stress, like UV light, can trigger it to pop back out and enter the lytic cycle.
And you mentioned earlier this integration can sometimes change the bacteria.
Yes, lysogenic conversion. Sometimes the prophage DNA carries genes for toxins or other virulence factors. So the phage infection actually makes the bacterium more pathogenic. Diphtheria toxin, cholera toxin, and those are encoded by prophages.
So just to be clear, prophage is integrated phage DNA in a bacterium. Provirus is integrated viral DNA, usually from a retrovirus, in an animal cell.
Perfect distinction. Same concept, different kingdoms.
And this leads to the idea of latency in animal viruses, like herpes viruses causing chickenpox, then shingles years later. How do they hang out dormant?
With herpes viruses like varicella zoster virus, VZV, the viral DNA doesn't usually integrate like HIV. Instead, during latency, the viral DNA persists in the nucleus of nerve cells, often as a separate circular piece of DNA called an episome.
So it's not actively making viruses, just waiting.
Exactly. It's mostly silent. Then, things like stress, aging, or immunosuppression can trigger it to reactivate, start replicating again, travel down the nerve, and cause a shingles rash.
Okay, let's talk about a virus that's constantly changing. Influenza. How does it get into our cells, and what are the two ways it evolves that keep us chasing it?
Influenza uses its surface spike protein, hemagglutinin, HA, to bind to sialic acid receptors on our respiratory epithelial cells. That's the key to get in.
And then it changes its HA.
Constantly. Two main mechanisms. Antigenic drift is the slow, steady accumulation of small changes, like point mutations, in the genes for HA, and the other main surface protein, neuraminidase, NA.
That's why we need a new flu shot every year.
Precisely. Drift happens all the time, creating slightly different strains that our existing antibodies might not recognize as well. So we update the vaccine annually to try and match the circulating strains.
And the other mechanism, the big one.
Antigenic shift. This is much more dramatic and abrupt. It happens when two different influenza viruses, say a human flu virus and a bird flu virus, infect the same cell, often in an intermediate host like a pig.
And they swap genes.
Yeah, their genomes are segmented, like they come in different pieces. Inside that doubly infected cell, the segments from both viruses can get repackaged into new variants. If a new virus emerges with, say, a bird HA that humans have never seen before, combined with human flu genes that allow it to spread easily between people.
That's a pandemic waiting to happen.
That's exactly how pandemics arise. Antigenic shift creates completely novel subtypes that bypass most pre-existing immunity in the population.
Okay, shifting to how we detect these viruses in the clinic. Let's compare the main methods. NATs, antigen tests, serology. When would you use each?
All right. NATs nucleic acid amplification tests, like PCR or RT-PCR, they detect the virus's actual genetic material, its RNA or DNA. They're incredibly sensitive and specific.
So best for early diagnosis.
Absolutely. They can often detect infection very early, sometimes even before symptoms appear or when the viral load might be low. They're the gold standard for confirming an active infection, like we saw with SARS-CoV-2.
Then the rapid tests, antigen tests.
Right. Antigen tests, often those lateral flow assays you can do at home or point of care. They detect viral proteins, like the spike protein or nucleocapsid protein. They're fast, cheap, really useful for quick screening.
But less sensitive.
Significantly less sensitive than NATs, especially early in infection or later when the viral load drops. You need a fair amount of virus present for them to turn positive. So a negative antigen test doesn't definitively rule out infection, especially if someone has symptoms.
And finally, serology. Looking for antibodies.
Serology looks for the host's immune response to the antibodies your body makes against the virus. It tells you about past infection or vaccination status. It's not useful for diagnosing an acute current infection because it takes days to weeks for detectable antibody levels to build up.
So NAT for early confirmation, antigen for rapid screening, serology for past exposure.
That's a good summary. Oh, and historically, we also use cytopathic effects, CPEs, looking for visible damage viruses caused to cells in culture, like making them fuse together, syncytia. But that's slow and needs a specialized lab.
We have to mention prions before we wrap up. Not viruses, not cells, just protein.
Yeah, it's mind-bending. Prions cause transmissible spongiform encephalopathies, or TSEs, fatal neurodegenerative diseases like Creutzfeldt-Jakob disease, CJD. The infectious agent is simply a misfolded version of a normal cellular protein called PRPSC.
And this bad protein makes good proteins go bad.
Essentially, yes. The misfolded PRPSC acts like a template, causing the normal PRPC proteins in the brain to misfold into the abnormal shape. This triggers a chain reaction leading to aggregation, neuronal cell death, and that characteristic spongy brain tissue.
Truly bizarre and terrifying. Okay, last clinical point. Phage therapy, using viruses to fight bacteria.
It's an old idea getting new life. Bacteriophages, or phages, are viruses that only infect specific bacteria. They're harmless to human cells. Phage therapy uses guadic phages, the ones that kill bacteria, to target infections, especially those caused by antibiotic-resistant bacteria like Pseudomonas aeruginosa or MRSA.
So, fighting microbes with other microbes.
Exactly. As antibiotic resistance grows, phage therapy is becoming a really important area of research and potential treatment. Hashtag tag outro.
So we've covered a lot of ground, huh? From the unique structures in fungi like ergosterol to protozoa using cysts for survival, helminths that don't multiply in the host, and then these complex viruses that hijack our cells and evolve so quickly through drift and shift.
Yeah, and the key takeaway for you as you prepare for the quiz in your future practice is that understanding these details really matters. Knowing about ergosterol helps you understand why endosols work. Knowing about cysts tells you about transmission risk. Think fecal oral precautions. It connects the basic science directly to patient care. Absolutely. It's about moving beyond just memorizing names to understanding the mechanisms. That's crucial for infection control, patient education, everything.
Right. And it leads to a final thought, maybe a bit provocative. We talked about multi-drug resistance in Candida auris, and it's a huge problem with bacteria too. But then we discussed phage therapy using viruses to kill bacteria. It makes you think, doesn't it? As our miracle drugs, antibiotics, start to fail more often, maybe one of the most promising paths forward involves harnessing these natural predators of bacteria, these viruses we've mostly ignored therapeutically for decades. Perhaps the next big weapon in our arsenal is actually viral warfare, but this time on our side. You might want to look into how phage therapy is actually making its way into clinical trials now. It's fascinating stuff.